Hello. Yeah. Mm. Okay. Welcome to week 11. Sorry. One second. Mm. Yeah. So, welcome to week 11 of Computational Systems Biology. My name is Shoma, and this course is taught by Professor Karthik Raman of IIT Madras. So, in the last few weeks, we've talked about different types of analysis techniques uh, for metabolic networks, including flux balance analysis and their variations, RFPA, room, and so on. And today we'll talk about mostly uh, gene regulatory networks, how to model them, and so on. So let's get started. Okay. So by definition, gene regulatory networks are basically network of genes and transcription factors and other elements involved in gene expression regulation. So basically, each of these nodes in the network represent a gene or a transcription factor or some other elements of the genome, and these actually interact with each other and regulate each other to ultimately keep the system in a stable state. Right. So the transcriptional regulation of genes underpins all essential cellular processes and is orchestrated by intricate interplay of many molecular regulators. And as I was saying, at the forefront of gene regulation are transcription factors with specific regions on the DNA called CRE, or which are known as cis regulatory elements. And these are mainly made up of promoters and enhancers. So these are the places where the transcription factors and the other elements bind and thus regulate the expression. So together, the interactions between transcription factors, key elements, and the genes form these gene regulatory networks, which govern cell identity and cell feed decisions and play an important role in the development and progression of various diseases as well. So variations in these genes, basically mutations or different types of variations in these genes cause can cause different diseases. And like if one of the genes is affected, and also for example a transcription factor is affected, but that transcription factor is involved in regulating five other genes, so most probably the expression of those five genes will also be somehow affected. Now, with the advancement of high-throughput omics, it is possible to profile the many molecular features involved in gene regulation. However, the reconstruction of these networks possess significant challenges that necessitate the development of powerful and efficient computational tools to unravel the regulatory interactions of gene regulatory networks. So, the first type of tools or model to use to model basically gene regulatory networks are logical or Boolean models. So basically, these are logic-based models. So these are basic and simple in terms of the model. These are basic and simple, and these are logic and discrete logic based. And it was introduced by Kaufman and Thomas. The reconstruction of regulatory network that controls the development of sea urchin embryo is a seminal example of profound insights that quantitative examination of regulatory network models can provide. Yes. Okay. Sorry. Mm. Yeah, as I was saying, so these are simple and logic-based models, and these are discrete models. So you can have values 0 and 1 and other classes of variables, but not decimal values or something like this. So these are discrete values. Either it is on or it is off. Cannot be something in the middle. So Yeah, so these logical models represent the local state of each entity in the system. So, say for example, you want to uh, represent the entity of a gene. So if it is getting expressed, you put it as one. If it is getting expressed, you put it, it is not getting expressed at all. You put it at zero. It is getting somehow in some other condition that it is following. You can put it in another class variable. So this yeah, so these logical models represent the local state of each entity in the system. For example, gene, proteins, and small molecules at any time level as a uh, discrete level, and the temporal development of the system is often assumed to be occurring in synchronous discrete time steps. So this is the assumption that goes into the Boolean or the logical models, that you always assume the temporal development of the system. So over time, the system only uh, uh, the development of the system only occurs in discrete time steps. So it's not a continuous variable, it's a discrete variable. And entity levels are updated at each time step, allowing according to regulation functions. So this is actually the figure shows a good example of a Boolean network and a Petri network. So on the left is a Boolean network. So you have these three elements, A, B, and C, and these are their logic states. So 0, 1, 0, and 1. So if you see in the first confirmation, it's 1, 0, 0. In the next confirmation, it becomes 110, and in the third confirmation, it becomes 111. Then it again moves to 011, then 001, then again 000, and the cycle continues. So these entities A, B, and C are existing in either 0 and 1 states, transitioning according to the regulation functions as given here. And these thin arrows actually are depicting regulatory relationships. And so basically, these arrows, the small arrows between the three elements, uh, basically the regulatory relationships between these three elements. But the arrows that are showing in the middle, so these arrows are actually telling time steps. So what happens after that? Right. So on the yeah, so if you write down the functions, regulatory functions, it becomes something like this. So if A and C both are zero, B is also zero. This is this condition. Then it's a zero, one, one. I think A is zero, B and C are one. I think it's this condition. Yeah. So if you want to write down the functions like this, it is very much possible. So this is a much more simpler version of what we actually model. Now, if you consider a little bit more complicated network, so this is called a Petri net, consisting of place entities and transitions, which are the regulatory functions, and these are connected by arcs. These dark blue tokens, they represent discrete values, and the transitions consume tokens from input places and produce tokens in the output places on firing. And transition activation occurs when input places contain sufficient tokens. And so basically, these are these blue dots. These are representative of tokens. So at the input, uh, they use up tokens, and at the output, they produce tokens depending on the output of the network. So yeah, this discrete modeling, all of this discrete modeling allows us to purely rely on qualitative knowledge. Such models can be analyzed using a broad range of well-established mathematical methods as well. So these logical models, or the Boolean models that we see, are actually very useful, and they make the model much more simpler. So if you have a system that you want to study, and you do not know much about the system, then a Boolean model or a logical model seems to be the correct approach, because not much information is required in that sense. Sorry. Okay, so coming to Boolean models, these are quantitative methods like ODEs and stochastic simulation, and these are used for studying cell dynamics when detailed mechanistic knowledge and kinetic parameters are available. However, the inherent noise in the biological data often makes precise parameter determination challenging, if not possible, impossible. Additionally, quantitative model analysis can be computationally demanding, leading to practical limitations. And in many cases, these abstract or qualitative models are preferred due to their ability to offer insights into the system dynamics in a reasonable time frame, where these actually Boolean and logical models come into play. uh, yeah. Boolean networks are common qualitative modeling frameworks for gene regulatory networks, and they are capable of describing various biological phenomena such as oscillations, multistationary events, switch-like behavior. Boolean networks are powerful tools for studying GR dynamics. So different conditions, in different conditions, a system will may behave like an oscillatory system or a multi-stationary system or a multi-stable system, multi-stable system having multiple steady states. Now, uh, all this information can be very easily modeled using Boolean models. And if you see literature around these, sorry, oh, okay. I thought somebody said something. Okay. So if you look at the literature around these kind of models, so there's a very famous there are actually there are a lot of works done on this. So basically, Boolean models, if you see in different systems. So basically, say for example, a cell is transitioning from one state to another, uh, and you want to model the whole dynamic thing. So Boolean models offer a lot of like ease of doing this modeling because of the less knowledge required, because we do not need this accurate parameters as in OD to actually describe the model. It's much more simpler, and you'll see a lot of papers. They have done a lot of work using these Boolean models. But again, since you are assuming the assumption there is, it's your system is either in a zero or a one state, right? So you do not have anything in between. So either it can be an off or a non state. But in practice, the biological systems that we see, they're not exactly that discrete. So they have, even talking about cell states, they have extreme cell states and they have also intermediary cell states as well. So it becomes much more challenging to describe those kind of behavior using Boolean models. But if you have a much more simpler system and you assume a lot of stuff around it, and then you do the modeling, then actually Boolean models will give you quite a decent uh, result. Okay. And each of these systems uh, actually, each of these types of behavior, oscillations, multi-stationary events, switch-like behaviors, and these are actually very well described using Boolean networks for studying GRI. Okay. So in a Boolean network, the Boolean variables represent the gene activation states, one for active, zero for inactive, and the Boolean functions determine the state of each variable based on the state of other variables. And these functions are updated periodically to simulate the GRN dynamics. And these Boolean functions can be represented as logic formulas or truth tables. And excuse me. Okay. Yeah. In a graphical representation, each of the nodes represent the Boolean variables, which are the genes, and the edge between two nodes will determine the relationship between the two nodes. It can be either a positive relationship uh, and or a negative relationship, right? So, and promotion and inhibition edges are typically distinguished by different types of arrows, but yeah, that is the idea. So yeah, this is one example of a Boolean model. So this is a graph representation of a Boolean network. And so for example, let us consider this graph. So what it is saying, okay, like it is much more abstract in nature than we think of. So this whole figure represents a network having four genes. So you have A and B here, C is here, and X is here as well. In which A and B have a positive influence on X, C has a negative influence on the same gene, basically X. So you have A and B upregulating X, and you have another gene C which is downregulating or inhibiting the same gene. This suggests that the formula expressing the condition uh, for the activation of X, there will be a positive occurrence of A and B, while where there will be a negative occurrence of C. This is because the truth of A and B favors the truth of X, while the truth of C disfavors the truth of X. Right. So it's not that hard. It's simple to understand. So you have A and B which are upregulating another gene X. So your uh, truth. So when A equal to one, B equal to one, or A or B equal to one, then X is equal to one. But for C, if C equal to 1, X equal to 0, and if X equal to 1 is C equal to 0. So it kind of is an opposing kind of a relationship. But this representation is very abstract. It does not specify the way genes influence each other. So uh, how much does the gene actually is involved in regulation, that is not specified. Yeah. Okay. So yeah, this is a little bit more detail of that same Boolean figure. So in all the previous Boolean functions, A and B have a positive influence of X, while C has a negative. And as a consequence, several different Boolean networks can correspond to the same graphical representation. So for example, uh, uh, okay, let me go through it, then I'll give you the example. So on the other hand, starting from a generic Boolean function, it is not always possible to obtain a clean and simple graphical representation. So basically what it is saying here is basically you can have different Boolean networks uh, which can correspond to a similar type of a graph representation, but the opposing thing, it is not easily possible to have a clean graphical representation from a generic Boolean function. While it is easy to do it for formulas in which the occurrence of each gene are either all positive or negative, for formulas containing a gene having both positive and a negative occurrence in the translation into graphical representation is not straightforward. This happens, for instance, in the second case. So basically, these are all different cases. So in the first one, you have A and B and not C. Uh, it can be A or B and not C. It can be A and not C or B. So it's the same graphical representation. Basically, A and B upregulating, C is downregulating, but the conditions may vary as you see it from here. Uh, yeah, so for the second instance, so A or B and not C. So all these actually conditions satisfy this particular representation. But the second condition, A or B and not C, uh, what it is representing is basically either A has a positive or a negative influence of X on negative influence on X depending on the activation states on of B and C. So so basically if you what it is saying is if you either have A or B and not C, you this is this actually the system works, the network works, X is uh, getting produced. So, but this is a very kind of a ambiguous statement because you do not know uh, uh, what is the influence of A or B on X actually, right? So, because you do not know the individual influence of these genes on the system, so that becomes a little bit tricky. So a possible solution is to create an extended graph which includes nodes representing combinations of genes rather than single genes. So that becomes much more informative. Oh, okay, so if you talk about the synchronous nature of these Boolean networks, so basically the dynamics of a Boolean network model is given as a sequence of steps in which the value of the Boolean variables are updated with the values obtained by associated Boolean functions. So usually two alternative updating schemes can be considered, which are one is the synonymous scheme, one is the synchronous scheme, one is the asynchronous scheme. So the synchronous scheme is deterministic, and all the variables are updated simultaneously according to the associated Boolean functions. And yeah, and this approach is based on discrete time steps and is computationally efficient and easy to implement. Synchronization of updates implicitly assumes that all biological events have a similar duration. So that is what I was saying. Like you make assumptions like these, so you need to respect these assumptions when you are um, kind of inferring the results. So these are actually kind of drawbacks of this kind of models, right? So synchronization of updates implicitly assumes that all biological events have a similar duration. This is often quite a strong assumption from a biological viewpoint, since in general the time scales of biological events can vary from fractions of seconds to hours to even days, months, and years. Right. So the variability is very high. So it's the assumption is very you can say wrong, at least. Yeah. To address this problem, we can make use of an asynchronous key, which assumes that only one variable can be updated in a single step, and every variable is equally likely to be updated. And in this approach, the nodes can be updated in any order, and the dynamics become non-deterministic. So the advantage is that you are almost mimicking a stable biological system, but the disadvantage to this is basically you are updating only one variable uh, uh, in in one iteration. Here you are updating one variable in one iteration, but the thing is the solution of the model, or the result of the model, they can become non-deterministic because there is no guarantee that the solutions will converge like this. But synchronization, if is happening, it, if you assume synchronization, there is a much greater chance of having a deterministic solution compared to an asynchronous condition. So the idea is simple. In synchronization, you assume the time scales are same across all the processes, and in asynchronization, you only assume that in one iteration, only one process can be updated. So that is the only difference between them. But yeah, in terms of assumptions, they vary a lot. So the kind of results that you get, you have to justify those results on the basis of the assumptions, right? So that becomes a little bit more trickier. Okay, so next we come to the concept of attractor analysis. So basically, what is it? Is so assume you are given an initial configuration of the Boolean variables. The model evolves following either a synchronous or an asynchronous updating scheme, and this causes the values of the variables to change, describing new configurations reached by the modeled GRL. And after a number of steps, the model typically reaches a state known as an attractor. So what is this attractor state? It can be a self loop, which is a single state attractor. It can be a simple loop in which a cyclic sequence of states is present. And also it can be a complex loop which is an alternation of two or more simple loops. Right. So when the synchronous update scheme is adopted, it is only possible to reach self loops and simple loops since the dynamics is deterministic. Right, as I was saying, in the previous slide only, so if you assume synchronization, your solution will have deterministic properties, but and so the attractor states it is possible to reach are only your simple loops and your self loops. And the simplest type of attractor, which is a self loop, corresponds to a fixed point in the Boolean function of networks. All the updates do not change the values of the variables since the Boolean functions are computed independently from each other. A self loop is the same regardless of the chosen updating schemes, which is either synchronous or asynchronous. But in more complex networks, the updating scheme influence the probability to reach the same attractors where attractors reached in the synchronous models are not present in the corresponding asynchronous model. So what it is saying is basically depending on your updating scheme, synchronous or asynchronous, obviously the solution of the model will change depending on it can become non-deterministic if it's a asynchronous process. But the idea is that you can generate these very specific type of attractor states for asynchronous model compared to synchronous models. So in synchronous models, can get these self loops and simple loops, and the dynamics will be deterministic. But in a asynchronous case, uh, uh, it is more possible to reach complex loops, basically switching between two different loops, and the update schemes actually influence the probability to reach the same attractors, where attractors is biologically very relevant because they are not often correlated to the gene activation configurations of specific cellular phenotypes. Uh, Besides, analysis of attractors helps us to understand the system dynamics and compare it with experimental data. And also, detecting attractors is sometimes challenging because, particularly when the initial set of configurations are unknown, uh, large Boolean models are studied. So at that condition, the detecting the attractor becomes much more complicated, uh, and much more computationally difficult. Okay. So as regards the first aspect, the problem is that it requires testing the model by considering possible 2 to the power n configurations, where n is the number of variables in the network. So this is a very computationally exhaustive process. An approach to overcome this problem is to sample a larger number of initial configurations and calculate the probability to reach certain attractors. So the idea is that you are testing for a fact key. You have a model created. Now you want to see, depending on your updating scheme and what updating scheme you are doing, what kind of attractor state is it reaching, and that can be biologically very valuable to understand. Right? Okay, uh, just a second, I'll be back. Just give me 2 minutes. Ah, yeah, sorry for that. Okay. So, we have discussed Boolean models, how they work, what kind of updating schemes can lead to different types of solutions of the models, and what in general the model represents in a graphical setting. Now we'll use that as an like use that all this information to actually use that in the modeling of human MTB interaction. So MTB is a bacteria which causes tuberculosis. So we'll discuss about how we can model this whole process using the conditions that we talked about and get a understanding of the system. Yeah. So the first step of it is uh, basically the network construction. So basically a comprehensive network model of host pathogen interactions in tuberculosis infection is to be constructed using extensive literature data. We have discussed how to do this kind of stuff earlier as well. Okay. Now the model incorporates both innate and adaptive immune responses because our body, so the response of our body to such an infection is not going to be very simple. So it's going to be starting off with a set of immune response. Then after a few days, when the antigen specificity has been established, certain other certain other immune functions will be activated, so antibodies will be produced, and then it'll ultimately be removed from the system. But it's not a very simple process. So there are various processes that occur depending on the different conditions of the antigens processing, right? Okay, now in our case, the model should incorporate both innate and adaptive immune responses, as well as various aspects of mycobacterial virulence and defense mechanisms. As I was saying, so these are complicated processes, so you have to almost incorporate every thing known about these kind of things. Right. Yeah. So the model aims to provide a systems level understanding of these interactions and addresses critical questions regarding immune response factors and bacterial defense mechanisms. So if you construct the network, it will have 35 components, which are including 56 of them are host components and 18 are bacterial virulence factors. So you have to understand the whole context. So the bacteria will have certain proteins that will be interacting with the human cell receptors, and human proteins. Some of the human proteins will be also interacting with the uh, bacterial cells and receptors and other signaling components there, right? In this case, the model has 56 host components and 18 bacterial components. Yeah. The host components span molecular, cellular, and cellular state levels, while virulence factors are specific to the bacterium. So, yeah, so I put the table in the next slide. So this is actually the whole network. Looks very complicated, but yeah. So I think you can like uh, recognize these cells, TH1, TH2, TH17, which are coming from this naive T cells, the TCs, CDR1s, then other types of cells involved in immune processes, eosinophils, basophils, mast cells, etc., etc., B cells at the end as well. So a lot of things are going on here, a lot of interactions and everything. So this is how a host pathogen network is represented in MTB. So the connections between the various nodes are indicated by the colored arrows. The black arrows indicate connections between processes that follow sequentially. Green arrows indicate activation, while red arrows are representative of inhibitory reaction. So yeah, it's very clear in that sense. Bacteria is indicated in darker colors. So these are, uh, uh, I think, yeah. So these are your bacterial elements. So the bacteria is uh, shown in darker colors. So you have, I think, mm, yeah, yeah. These are their, yeah. So basically, this is the whole network. So here is the bacteria anyway. Processes such as phagocytosis are colored as gray, while molecules such as uh, cathelicidin are all colored as cream. So you have these processes involved here, apoptosis here. Then you have some other things, bacterial clearance. NK cells, phagocytosis, etc., etc. Right. Cells like natural killer cells, Th17, and GD cells have been implicated only in vitro, and they have not been considered in the Boolean network, and all the box has been hatched. So this is the whole network that we are studying. So now, if you want to, one second. Yeah. Now, if you want to understand the events in the immune response system. So, first step is the entry into the host. Then it enters the host through the respiratory route in the form of droplet nuclei, and it resides inside the lungs. Then the bacteria can bind to various receptors in the host surface, such as T cell receptor, SPA receptors, etc., etc. And it interacts with these receptors to actually get entry into the cell. Then, after it is done, then it starts phagocytosing. So MTB sinks in or is engulfed into the host cell, depending on the receptor involved in binding. Then receptor-mediated signaling occurs, where basically signaling cascades are triggered upon the host infection, like TLR signaling, CR signaling, etc. So antimicrobial, mycobacterial molecules like cytokines, etc., are secreted because of this, because the body understands that there has been an infection. Right. So after this, the antigen processing takes place. So basically, a small part of the bacteria or the mycobacteria antigen, they're loaded into suitable presentation modules such as the MHC and CD1, and are transported to the surface of the cells, which are then recognized by TCRs and co-stimulatory molecules. Then T cell differentiation occurs after antigen presentation, and in this case, the naive T cells actually differentiate into TH cells and TC cells, helper and your cytotoxic T cells. And then upon infection, various cells of the immune system are also recruited, different types of white blood cells, different types of B cells, etc. And then this binding of the cytotoxic T cell triggers a pathway which induces targeted cell apoptosis. And this is done using this granulosin released by this cytotoxic T cells. And then apoptosis occurs. And after that, various cells of the immune system actually aggregate at the infection site to form a granuloma to contain the infection and prevents the dissemination of bacteria. So this is the whole idea, the whole procedure. So start off with the entry, then you go through all these different procedures, and then ultimately you the bacteria is killed off. Right? Now, if you want to do a Boolean modeling on this, right, on this whole set of information that you have. Now, yeah, so in your model, you'll have the nodes, and each node will represent a component's qualitative state, as we were discussing. Like it's a 0 or a 1, it's a on or a off, right? So these nodes will include also the system components and bacterial defenses, as we were discussing earlier as well. Now you have these transfer functions, you have logical operators and or not, etc., etc. So activation can be given by or, inhibition can be given by and not, and joint activation can be given as an and. And this uncertainty is handled by random variables drawn from random distributions, right? And then an asynchronous update method is adopted, and the cell states evolve iteratively based on the update rules, right? So these are two examples of intermediary events, interaction events. So reactive oxygen intermediates are formed during the infection procedure and all. And so this is actually released by the APCs, which are the antigen presenting cells under pro-inflammatory cytokine influence. And the bacterial defense components mitigate ROI's detrimental effect. So the Boolean function will be ROI equal to activated phagocytic cells and pro-inflammatory cytokines and not. So these are your bacterial components. So SOD A, SOD C, BPOP, whatever these are. So these are your bacterial elements. Right. So this is how you can like represent each of the logics in the model, right? So another example is a Th1 cell activation thing. So it produces IFN gamma and TNF alpha. These are cytokines involved in infection control. And these T cells differentiate into Th1 upon stimulation with IL-12, which is another type of cytokine. And macrophages increase the Th1 cell population in the presence of chemokines. So the function representing Th1 cells will be the presence of bacteria, obviously, and T cells and IL-12 or IL-18, because you need either of these to actually IL-12 or IL-18 to actually differentiate Th0 to Th1 cells, right? Or macrophage and chemokine signaling. So this is how representation of Boolean modules are done in network dynamics. So, coming to the example, so yeah, these are the different parameters that are considered for this Boolean network model. So you have the bacterial load, which you have the initial and the minimum and the maximum. You have you can have so these are actually parameters. These are not the actual unknowns. So these are parameters that you can either estimate, as in ODs, how it is done, and also in this case here as well. Right. So these are different parameter values that you assume or you estimate. So in this case, the assumption is based on the fact that assumption is based on the previous literature and also experimental data. Yeah, so bacterial load is one. Then you have parameters like bacterial growth rate, incremental change. So basically, how much is the volume of the bacteria increasing or decreasing, and the rate at which the population is increasing. Then you have the host clearance efficiency. So the host is also trying to clear up the bacteria. Then so intake of drugs can alter these particular conditions. So why I have given this example, such a descriptive example, is of because of to show you the fact the things that we need to consider, the kind of thinking that we need to have before doing such a modeling, because such a host number of things you need to consider before you actually write a model. Right. Yeah, and so host clearance efficiency is also plays a major factor. Delay factors for immune responses, because obviously, like, the body does not react at like a fraction of a second time scale. It takes some time. It takes a day or a two to actually understand what the infection has been, and then it deploys its actual immune response, right? So that is also there, and also onset delay of adaptive immunity, that is also there, because moving from your um, innate immunity to adaptive immunity, innate will be like uh, very fast, depending just after the yeah infection has started. So the innate is going to be very fast. Adaptive is going to take some time after the innate uh, has kicked in. So that is all. There is also a delay for that as well. Onset delay of adaptive immunity. Okay, so these are different parameters values that they have assumed, and what are the possible ranges, and also that they have mentioned here. Right. Okay, now coming to the whole simulation. So initialization sets initial states of components at infection onset, and the model evolution spans Tmax time steps, which is 105 in this case, corresponding to 15 weeks. So, like, what is uh, what Tmax time steps here means is 105 times you are asynchronously updating the model, right? So that is what means here, which actually corresponds to 15 weeks in terms of if you just convert it. Okay, so for simulation parameters for 100 runs, so these are your parameters that are assumed from this table uh, only. Uh, so the result comes out as active disease 15 for 15 runs, then 42 runs for clearance and 42 runs for persistence. So if you use this in a extended simulation, so if you use this is for 100 runs, so if you do it for 1200 runs, the the percentage is almost similar. So you have 13% active disease, 41% clearance, and 46% persistence. So yeah. Yeah, so this is a state map of the qualitative states of important nodes during a single simulation run. So these are your nodes, which can be different cells. So these are different cells. These are pathways also involved here. Uh, Cytokine concentrations also involved here. Different cell types also involved here. So uh, uh, it is representing in terms of on state, which is basically the blue, and the white state, which is the off state. Right. And so this is a map that highlights the delay in activation of adaptive immune response machinery. So if you see uh, IFN gamma, you see apoptosis, you see steps like Th1RC. So these are all involved in the adaptive immune process where the activation occurs later compared to the innate immune process. And this actually is the variation in the load system of the bacteria for default parameter set. And the bacteria enter persistence actually in this scenario. So basically, uh, it goes up, then it goes down, and then it reaches a stable steady state, which is the persistent state, right? In the second case, actually, the phagocytosis is disabled, so the bacterial load increases and it stays like that only. In the third case is a outcome of active disease due to disabling phagocytosis. Uh, and sorry, so this is the third one. I think it has come up in two comments, right? The fourth one is basically showing persistence due to disabling delay in adaptive immune response. So this is showing persistence due to delay of the adaptive immune response. And is the final one is basically showing bacterial clearance due to deletion of TGF beta. Okay, so basically these are the different models and the conditions under different conditions how these different things work. So this was a good example on that. And if you want to like kind of summarize this in one slide, is basically you start with the network reconstruction using literature on host and host tuberculosis interactions to identify components and interactions. In this case, it was 75 nodes with 56 host components, 18 bacterial virulence components, 11 cellular processes, 19 cells and cell states, and 26 molecules. Then you do the network assembly. Then you derive the Boolean transfer functions, initialize the node states, simulate the network, and then you have these three conditions possible, and what is the probability that the states remain in either of these three conditions. You get that answer. So yeah, so this is actually the example that I wanted to discuss. Okay, I thought someone was. Okay, so I hope you've understood how this kind of Boolean modeling works, and I hope I have given you an example to actually make you appreciate this particular yeah model. Now we come to a concept called robustness. So we have already talked about robustness and resilience in terms of biological systems, but in terms of Boolean in terms of Boolean networks and all, what does robustness actually mean? So it is a ubiquitously observed property of biological systems, and what it is basically is I'm trying to find the statement where it is written. So basically, I'll not read this out. try to explain what it is. So you have a system, and the system is in steady state. Now you try to move that system out of the steady state slightly. That does the system go back to the steady state, or does it move to a different steady state, or does not? It becomes unstable or whatsoever. Depending uh, on the nature of the system, on to go back to its original steady state, that is what is known as robustness, right? And this robustness facilitates evolvability, and and robust states are often selected by evolution. Such a mutually beneficial process is made possible by specific architectural features observed in robust systems, right? We are coming to that only. The architectural features observed in robust systems. But there are trade-offs between robustness, fragility, performance, resource demands, etc., which explain system behavior, including patterns of failure. Insights into inherent properties of robust systems will provide us with a better understanding of complex diseases and a guiding principle for therapy design. Right? Yeah, so basically we were talking about attractor states. So if you move the system here to here, and then it moves back to its original adaptive states, it's a robust adaptation, returning to a periodic attractor. Now, if it moves away and becomes unstable, it's not robust, and so on. So uh, now we'll talk about system control, yeah, using feedback. So this robustness is something that is related to the system control, because uh, in system control, you basically want the system to stay in a stable state for a longer period of time. So, system control in biological regulatory networks, such as those governing cell cycle, circadian clock, etc., on relies on both positive and negative feedback mechanisms to achieve this robust dynamic responses. Negative feedback, particularly, is an integral feedback, because I don't know if you remember or not, we talked about negative feedback and how it can form these oscillations and so on. Yeah. Negative feedback, particularly integral feedback, is a fundamental mode of control that enables robust adaptation to perturbations, as exemplified in bacterial chemotaxis. Integral feedback ensures perfect adaptation by maintaining responsiveness to a wide range of stimuli. On the other hand, positive feedback actually amplifies the stimulus, leading to bistability and robust pattern formation. This is a very good example, actually, for this, which is the Drosophila segment polarity formation. It's a negative feedback loop which forms this robust pattern. Computational models have demonstrated that positive feedback loops are essential for reproducing experimentally observed and patterns and ensuring robustness in biological system. Positive feedback also plays critical role in signal transduction, cell cycle, self-fed decision processes, etc., contributing to switch-like behavior and enhancing the robustness against noise and fluctuations in stimuli. So in case of multi-stable states, it acts like a switch-like uh, uh, it acts like a switch-like behavior. So you have multiple states which are stable. It can switch between these states seamlessly. And the combination of these negative and positive feedback mechanisms allow biological subsystems to perform their functions effectively while maintaining robustness. Right. So there comes the important concept of what do you need to do to keep this robustness intact in a system. So here comes the concept of redundancy and diversity, right? So alternative or fail-safe option. So the concept of alternative or fail-safe mechanisms enhances robustness by providing multiple means to achieve a specific function, which can compensate for failures in one component with others. This mechanism encompasses redundancy, diversity, and overlapping functions, offering flexibility and adaptability. While redundancy encompasses identical or similar components replacing each other, diversity relies on heterogeneous components with overlapping functions. One such example is a gene duplication, followed by specialization or extensive gene loss, serves as a crucial mechanism for evolutionary innovation. At the network level, uh, Examples like glycolysis, oxidative phosphorylation, they demonstrate alternative pathways adapting to environmental changes, showcasing phenotypic plasticity as a part of robustness. So, I'll not read through the entire thing, but the idea is that if you have redundancy or if you have similar elements in the system that can perform, sorry, different elements in the system that can perform a same job, that actually contributes to robustness and diversity, right? So diversity is very important where you have an alternative or fail-safe option where basically if a particular gene is knocked out, some other gene can actually take up the function of the gene. It is not possible all of the time, but in case of housekeeping genes, it is the case. So you have a lot of redundancy in these biological systems, and this diversity actually helps keep the system robust, not moving away from its steady state. Right. So there comes two concepts of modularity and decoupling. So what is modularity? If you remember in the initial graph theory classes that we talked about, modularity is basic identification of a connected set of nodes which have a similar type of behavior, right? So modularity is a fundamental concept observed in across various biological processes. It serves as an effective mechanism for containing perturbations and minimizing their effects on the whole system. It encompasses physical, functional, spatial, and temporal modules, each contributing to the system's robustness and versatility. Examples include cells, which form basic units of multicellular organisms and interact with their environment and other cells, as well as hierarchically organized structures such as tissues and organs. So, what it is saying, like the functions and of the system are actually modulated in concentrated areas. So you have these separate modules inside the system which actually take care of these different functions. And the concept of decoupling is basically to isolate low-level variations from high-level functionalities. For example, molecular chaperones, like this is a very important one, heat shock protein 90, which not only correct misfolded proteins but also buffer genetic variations, allowing for adaptation while maintaining uh, the genetic diversity. So feedback controls are seen in bacterial chemotaxis, Drosophila melanogaster segmentation, repolarization segmentation, compensate for changes in network interactions or initial states, mitigating the impact of mutations. So bistability created through positive feedback can decouple molecular fluctuations from a system's committed state, enhancing a robustness to genetic and environmental perturbations. So then we come to the final concept. So all of this robustness, resilience, modularity, decoupling, everything is actually part of the network architecture, right? The concept of robustness is exemplified in modern airplanes through the design of their automatic flight control systems, which is AFCS. So basically, this is actually an analogy to robust systems, how robust systems should behave and how a robust systems designed to behave. So this system, automatic flight control system, what it is basically maintaining aircraft's flight path against atmospheric disturbances by automatically correcting deviations from the defined path. It achieves this through feedback control, utilizing the aircraft's flight control surfaces and propulsion systems. Now, and robustness is ensured by incorporating redundancy, as I was talking about in the previous slides, redundancy and heterogeneity into the AFC system. The redundancy is achieved by employing three modules with identical functions. So, as I was saying, the analogy is that having multiple genes doing the similar function. Three modules having identical functions, ensuring that failure in one module does not actually compromise the system's performance. Heterogeneity is introduced by designing each module differently from to prevent common mode failures as well. Similar system design. Digital technologies are utilized to implement these mechanisms, decoupling low-level voltages from digital signals to prevent noise from interfering with system functions. While this example pertains to man-made systems, the principle of robustness actually extends to biological systems as well, highlighting the fundamental organizational principle of evolving dynamic systems across various domains, including evolution, competition, market niches, and society. So the idea is that robustness is not only confined to biological systems, it is everywhere in the world. That robustness is something that we try to achieve, and we design our systems in taking inspiration from these biological systems to how to keep a system robust. Right. So yeah. Uh, so ultimately, we come to an architecture which actually uh, okay. Ultimately, we come to an architecture which actually describes this whole robustness procedure. Which is known as the bow tie architecture. So the idea is that you have big elements on top, big elements on top, bottom, and you have a neck of the bow tie which actually connects the top and the bottom elements, and those are the most important parts of the architecture. Right. So a bow tie architecture where many inputs are fed to a central core that is the unutilized to produce many distinct outputs is believed to contribute to robustness in many biological networks and may provide an alternative explanation for the rare yet devastating fragilities of the biological systems. In metabolism, bow-tie architecture provides formal description of large fan-in catabolized nutrients that produce a small number of activated carriers, which are ATP, NADH, etc., and precursor metabolites, which are present in the knot of the bow tie, that fan out in the synthesis of numerous building blocks. Example, nucleotides, uh, sugars, amino acids, and eventually larger macromolecules. This architecture provides exceptional robustness towards variable nutrient inputs through many degenerate metabolic pathways, again, redundancy there, that can be used to produce a few common building blocks. A simple example of this degeneracy is glucose metabolism can take place either by glycolysis or through the pentose phosphate pathway. Although these pathways can substitute each other if necessary, the sum of their metabolic effects is not the same always. Right. So I think that concludes my presentation. We will move into now the assignment previous year assignment questions. If you have any questions regarding this set of slides, you may ask, or we will move into the questions. Yeah, I think then we can move into the questions. I'll keep the questions here on the screen for a minute or two, then we will discuss the answers. Oh, sorry, by mistake. Stop sharing. Okay, you can, if you have the answers, you can write in the chat box. Okay, the answer is 2 to the power 5. So the answer is actually 2 to the power n. If you have 5 nodes, it's going to be 2 to the power 5. If you have n number of nodes, it will be 2 to the power n. So yeah, it's simple. Moving on. Okay, the question given here is you have G3 star, basically with G3 is expressed either from G1 or G2, and G1 is expressed with if the condition is G1 star equal to not G1. So the question is which of the following are true? So G3 is expressed when both G1 and G2 is repressed That is not true. Either of them have to be expressed to get G3 expressed. G3 is never expressed if G2 equal to 1 at all instants of time. Which is not the case. If G2 equal to 0 at all instants, G3 is expressed with Gi. No, but Gi has to be expressed for G3 to be expressed. So none of the above is the condition for answer for this. And if G1 equal to G2 equal to 1, then these both are active. Then the state of G3 in the next instance becomes 1. Okay. So it's yeah, it's quite simple. If you have any questions to ask, but yeah, moving on. So the answer is basically you have maximum for Boolean, then you have RFPA, then you have ODE, then you have SSA. Yeah. So Boolean model will give you the maximum. RFPA is a similar concept, but uh, so basically RFPA will be getting better than ODE, and finally is the SSA. Okay. Yeah, the answer is ODE will be the maximum if you want to model the dynamics, because it models it in like dt time, which is basically a very small instance of time which you can integrate over the time to get a fully analytical solution of the model using ODE. So it will give you the most. RFPA will give you slightly lower than that. Boolean, uh, in terms of dynamics, will give you very less because it can be either on or off. Like so, in terms of uh, ability to model the dynamics of the system, ODE will be maximum and Boolean will be the lowest. Okay. The answer is redundancy because it's the same. Sorry, it's the same gene getting duplicated. It's the same function to of the same gene. So it's basically form of redundancy, but it contributes to robustness. Okay, so positive feedback will bring in bistability and negative feedback will bring in noise and elimination. So yeah, moving on, I'll be back. The answer is modularity. So basically, you are separating out different modules inside the system. So the time scale in the process, difference in time scale process will different lead to different modules in the system. Yeah, I have one more question. So the answer is in a bow tie architecture, what is there is the core process, the neck, the processes in the neck, those are heavily conserved, highly conserved, and the different input processes. So you have a lot of redundant input processes on the top, and a lot of input processes, different input processes can give you the same output. That is the whole idea of bow tie. Okay, so that's it from my side. If you have any questions, you can ask, or we will close this session. Okay, then. Thank you for joining. See you next week. Thank you. Yeah, this recording will be uploaded on YouTube. No, not to worry. Yeah. Thank you.